For centuries, we have thought that we are somehow above the rest of the environment. However, in the past years, we've come to understand that we are still just a part of a huge ecosystem in which species are independent because of the established complex relationships between them. Looking into the whole human microbiome, namely those microbial communities inhabiting our gut, mouth, skin, and other body parts, we can see that, in fact, our own cells constitute only a minority of the total number of cells in the body. These microbes that live in our bodies and all around us have an impact on our health and can also cause diseases. It is a process that we don't yet fully understand. Of course, there are also other factors that affect us and our health. The rapidly changing world brings many challenges to which the human body has not yet had time to adapt. The progress of our civilization is an undeniable fact. However, we still don't have the tools to prevent and cure ailments and diseases that come along with it. Thus, we need to keep looking for solutions through scientific research, fueled by the development of new measurement techniques.
The One Health approach consists of large-scale collaborative efforts of multiple disciplines to attain optimal health of people, animals, and our environment. Our contribution toward the One Health approach is through the study of the exposome. The exposome can be described as all of the external and internal exposures that an individual encounters throughout the life. It includes chemicals, physicals, biological, and social factors that may influence human health and well-being. It can be, for example, place of living, amount of sun, pollution, stress at work or home, diet, physical activity, etc. How can we approach this holistic perspective? One way is through the use of multi-omics technologies to collect data regarding the different biological phenomena that are invisible to the naked eye.
One of the fathers of the modern synthesis of evolutionary biology, Theodosius Dobzhansky, once said, "Nothing in biology makes sense except in the light of evolution. Every biological phenomenon, at any scale, is the result of millions of years of evolution." Thus, if we truly want to understand any living being, we have to look back and see how it originated and changed over time to achieve its current form.
Genomes are extremely long strings built from 4 nucleotides represented by 4 letters, A, C, T's and G's. But how these 2 letters build a human? How do they do it to produce the magnificent diversity of complex organisms we see around us? Understanding this is one of the main challenges of the current century in the field of biology. The key to this question relies on the gene rotary networks, namely the complex network of elements that interpret the genome in a unique way in each cell, allowing them to produce different cell types that build different organs and that do different functions. Our approach to understanding how genomes produce complex organisms is through the study of how different elements of the regulatory network emerged, evolved, and contributed to the rise of new characteristics in different animal lineages. For that purpose, we use multiple omics techniques, each of them providing us a window into a distinct layer of the gene regulatory network.
Until recently, it was very difficult or sometimes outright impossible to study the diversity of all the biomes of the human exposome. The game changer was the development of next-generation sequencing technologies. They allow us to reveal the genome sequences of all species present in a sample, even those which were previously unknown.
What is more, thanks to AI-based methods recently developed in Małopolska Center of Biotechnology at Jagiellonian University, we can not only say who they are, but also what they probably do, and which species prefers to hang out together. From gut microbiome perspective, we can now provide a unique understanding of its composition and how it is connected to the health status. For example, in a recent study, we have shown that we can predict the COVID-19 patients' outcomes at the time of admissions to the hospital, based on their gut microbiome composition. But in our work in Małopolska Center of Biotechnology, we also look from another end of the exposomal concept. Since 2020, Krakow is taking part in Global City Sampling Day, organized by the International MetaSap Consortium. In this action, we collect samples from surface and air in public transport to better understand what lives around us and how it affects us. In a recent study stimulated by our work, it was shown that low-cost sanitation based on the probiotics Bacillus could effectively replace the widespread use of the chemicals, foregoing the risk of the further exacerbating AMR, antimicrobial resistant strains and pollution, and at the same time, leaving undisturbed the non-pathogenic environmental microbes. Today, antimicrobial resistance is one of the biggest challenges facing modern medicine. According to recent UN estimations, it will reach a level of up to 10 million deaths annually till 2050 due to AMR. Thus, we need to act now.
Evolution, with the basic principle of survival of the fittest and trait inheritance, has shaped all life forms on Earth, and we can see it acting in real time, such as in the arms race between pathogens and their victims, or as we have recently seen between humans and viruses. Often, in biological research, we need to use experimental models that allow us to generate data and test hypotheses. The most commonly used models in genetics are fruit flies and mice. Important features of experimental organisms are that they should be easy and cheap to read in the lab. That's why insects are such good models, and therefore we use them in the lab. Insects also have one of the most fascinating features of the animals in terms of gene expression regulation, which is the metamorphosis, or how one genome can build 3 different body types. The caterpillar, that's like a worm, the chrysalis, with the insect closes itself while gets ready to become an adult, and the adult, which has the final body shape of an insect. In a recent publication, we show that a single master regulator gene in the embryo evolved acquiring the potential to determine whether the insect will hatch as a juvenile stage that looks like an adult or as a larva looking like a worm. This is a clear example of how a very extreme trait can emerge in evolution by a single gene being on or off without the need for new genes. This basic research, attempting to understand the general mechanism of life and using large amounts of omics data, allow us to comprehend the causal connections between the genetic material and the characteristics of an organism.
Now, we want to go a bit further and investigate how temporal but large migration is affecting the city microbiome in context of appearance of pathogens or antimicrobial resistant strains. Our studies are in line with the recent UN discussion about the global early warning system, which will utilize the characterization of the city microbiome to biosurveillance of AMR and pathogens.
The studies of environmental and gut microbiome are extremely interesting on their own. Even more interesting are the interactions of these complex microbial communities with humans and their organs. These interactions are happening through metabolites, and a number of communication paths have already been identified, such as the ones between the brain and the gut, or the gut and the lungs, and so on. When it comes to cardiovascular diseases, quite surprisingly, all of their known risk factors, including well-known classical risk factors such as hypertension, hypercholesterolemia, smoking and aging, and novel risk factors like environmental stressors, including environmental pollution or noise and unhealthy lifestyle, have a detrimental effect on the internal layer of cells lining our cardiovascular system from the inside, called endothelium. Therefore, endothelial function measured in an individual represents an integrated biological index of the body's response to all cardiovascular risk factors.
Endothelium is a single layer of endothelial cells covering entire cardiovascular system, which was previously thought as a not important inert layer. Today, endothelium is recognized as a multifunctional organ because it regulates a number of important functions. For example, endothelial cells regulate vascular permeability, vascular tone, intravascular thrombosis, inflammatory processes, and many others. When these various endothelial functions are preserved, cardiovascular system is healthy. However, when these functions are impaired, endothelial dysfunction develops that promote inflammatory thrombotic responses leading to cardiovascular diseases. Endothelial dysfunction is a hallmark of multiple diseases, not only of cardiovascular diseases, but also has an important role in multiple non-cardiovascular diseases. Endothelial function determines also healthy aging. As Rudolf Atschul many years ago wrote, one is as old as one's endothelium. The mission of JSET, Interdisciplinary Research Center at Jagiellonian University, is to foster interdisciplinary studies in endothelial and vascular biomedicine.
At JSET Laboratories, an array of experimental methodologies have been set up to understand the mechanisms of diseases linked to endothelial and vascular pathologies, and to advance pharmacology of endothelium. The far-fetched aim of basic research at JSET is to translate this knowledge to humans, to better diagnose and treat endothelial dysfunction in patients with various diseases. Today, some methodologies to diagnose endothelial function are available and are used in clinical research studies of the JSET team in collaboration with the University Hospital in Krakow. However, they have limitations to be widely used in everyday practice. In experimental work at JSET Laboratories, a number of methodologies are used to understand why endothelial function goes wrong and how to reverse it back to its proper function. Starting with the unique magnetic resonance imaging (MRI)-based approach that allows to detect and quantify the degree of endothelial dysfunction in vivo in murine models of human diseases. To understand the mechanism of the impairment of endothelial and vascular function in various diseases, comprehensive studies on functional, biochemical, proteomic, molecular and metabolomic levels are performed. They are performed in vivo, in isolated organs, in isolated vessels and in cultured cells. To give one example, functional metabolomic assays are carried out, which are supplemented by metabolomic analysis, untargeted and targeted, to understand the changes from healthy to unhealthy endothelium and vascular system. Direct measurement of the altered balance of nitric oxide and free radicals production by EPR spectroscopy represents another important asset of JSET's array of methodologies to assay endothelial function, because unfavourable alterations in nitric oxide and free radicals production represent a hallmark of endothelial dysfunction across various diseases. Quite surprisingly, biochemical content of vascular wall or blood cells can be studied by Raman spectroscopy, making a novel use of this analytical method in JSET to explore biochemical alterations in endothelial cells or vascular tissue. Analysis of the organ injury by histological methods is an important endpoint of any pharmacological studies carried out at JSET Laboratories, because any disease is associated with altered microscopic structure of the organ. All these interdisciplinary methodologies at JSET are dedicated to understand mechanisms of endothelial and vascular dysfunction in various diseases. Coming back to the concept of exposome of modern world that promote various human diseases, this is also due to the deterioration of endothelial and vascular function by our unhealthy lifestyle.